All right, this is our final presentation, the bioinformatic and experimental investigation of our given gene. In this project, we are working with Brachypodium. It is a model organism because it's been fully sequenced, but not all of its genes are identified yet. Our first step was creating a working map. We did this through an ab initio model, and we generated this through fGanesh. And this predicted that we had one gene from our genome and 8 exons, and it was on the positive strand. Then we did a homology-based approach using NCBI BLAST. We wanted to confirm our predicted model from the ab initio model. Using this, we found 4 express sequence tags that were derived from our matching cDNA clones. We had 4 matches. 1 was a 100% match, 2 were 82.26%, and 1 was a 78.12% match. This is our gene browser, and it shows the relative locations of those ESTs that we found in our last slide. This shows also that we had a high RNA sequence coverage. And this is also the browser that we use to later find our amino acid sequence. This is our phylogenetic tree. We used our phylogeny analysis to determine that our protein is a scar-like protein or an isoform of that. And we know this because you can see our gene's top glass match is in blue, and it was 100% match.
Another method we used to understand our gene's function was assessing its mutations. So another thing we did with our phytosome browser was assess the different mutations that were throughout our genome. And so to confirm that mutation, we designed primers that covered our suspected severe mutation that was at position 5133, which would create a stop codon. So we created primers that amplified the 743 base pairs surrounding that mutation. But our following Sanger sequencing results did not provide sufficient data to confirm its presence. And based on our phylogenetic tree and the phytosome BLAST results, we wanted to look more closely at the related organisms of Brachypodium in order to learn more about the genes related to the SCAR2 protein. And there's research showing protein homology between their Arabidopsis thaliana. So through a literature search, we found that it was involved in cell-to-cell adhesion, structure, and shape. So our research informed us that our gene was a part of the Wave complex and had an unknown involvement in actin polymerization. So it activates the ARP2/3 complex, but the regulation aspect of it is still unknown. And on the left, you can see that a mutated version of the distorted 3 which is related to the SCAR2 protein, causes trichome distortion. So you can see in images B, D, E, and G, the morphological phenotypes that were differing based on mutated DIS3 proteins. So using EFP browsers and data from Sam Hazen's lab, we assess the gene expression of our gene to narrow down where and when our gene is most active. So as you can see in the above images, there's high expression in the germinating seeds, young seedling, and the middle length grain. And this is all in the early stages of plant growth between 10 and 42 days after germination. So our hypothesis was developed knowing our genes involved in actin movement. We hypothesize it plays a role during initial plant growth, and this could affect plant development and cell shape, explaining its presence in the early stages of plant growth. Based on that hypothesis, we thought to use a growth inhibitor like caffeine as our experimental treatment, seeing as our gene seems to be most actively expressed during high periods of maturation, division, and elongation. We thought it would help us refine our gene's function by seeing if a growth inhibitor would reduce the expression of our gene. So to measure expression, we will quantify the data from the sample of each plant's genetic material 3 hours after treatment. So we did 3 plants with just a soil soak of water, and then our treatment was a soil soak with a 25 milliliter solution of 8 millimolar caffeine solution. And so we did some statistical analysis of these the gene expression. So these are the average CT values across the samples between the control, the treatment, and then samples provided to us that were wild type and mutant. And our original experimental design was to assess the seeds of the plants, but That didn't work out because of the seeds did not grow well. So we think that we would have gotten better results if we were able to look at the earlier developing plant. This was a small seedling.
So to summarize our results, what we do know is that our gene is part of the Wave complex subunit. From knowledge about the WASP and the SCAR protein are two similar protein families, and they both bind to the P21 subunit of the ARP23 complex. The effect of nucleation activity is still unknown, and the true function of our gene is also unknown, but we know that it plays a dynamic role in the ARP23 mediated cell migration, and we know that it has a large role in cell growth and the development of the cell shape. So that is what we know so far about our gene.
Thank you.